Welcome to another episode of Catch This. My name is Trudy Ray. Have you ever wondered what would happen if you were infected with two different viruses at the same time? A recent study aimed at addressing this question has produced some astounding new findings. Don't go away. The authors of the study wanted to observe the interactions between respiratory syncytial virus, or RSV, and influenza A virus, or IAV. So they infected lung cells with either virus or a mixture of both viruses. An initial experiment comparing the replication kinetics of each virus in co-infected cells to those infected with either virus showed that co-infection had no impact on the replication of IAV, but did lead to reduced replication of RSV. IAV and RSV each localized to distinct cellular regions during their individual course of infection. RSV aggregates in cytoplasmic complexes known as inclusion bodies, and IAV scatters more diffusely throughout the cytoplasm. Analysis of infected cells by fluorescence microscopy using antibodies against both the RSV and IAV nucleoproteins revealed that co-infection did not alter this localization. RSV was still in inclusion bodies, and IAV was still diffuse. The authors then analyzed later stages of infection when in single infections, these viruses assemble in structures called lipid rafts in the plasma membrane. Their results using antibodies against the virus surface glycoproteins, the HA protein of IAV and the F protein of RSV, revealed that in co-infections, both viruses were also simultaneously in the same region around the plasma membrane, suggesting that viral particles budding from the cell surface could contain components of both RSV and IAV. Using high-resolution confocal microscopy and a technique known as cryo-electron tomography, which reconstructs a series of image slices to generate a three-dimensional structure of a sample, the authors found that co-infection of cells produced two types of particles. The first type, called pseudotyped viruses, consisted of RSV particles with IAV glycoproteins. The second type, designated hybrid virus particles, or HVPs, were true hybrids containing the genomes and the surface glycoproteins of both viruses with distinct structural regions characteristic of each virus. Because glycoproteins determine which cells and cell surface proteins viruses can bind to, a concept also known as antigenicity, it is reasonable to assume that HVPs would have a modified antigenicity relative to IAV and RSV. IAV entry into cells occurs via the viral HA protein, which binds the cell surface protein sialic acid. RSV entry is infected in part via its F protein, which mediates fusion of the virus with the host cell membrane. To determine whether the HA and F glycoproteins on the HVPs were altered in terms of their antigenicity, the authors carried out a neutralization assay, which reveals whether an antibody can bind a glycoprotein and inactivate the viral particle. Anti-HA antibodies neutralized HVPs about threefold less efficiently than viruses collected from cells that had only been infected with IAV, suggesting that the HA on HVPs is different enough so that antibodies won't recognize it. In contrast, anti-F antibodies neutralized HVPs about as well as they did viruses isolated from cells infected with RSV only, suggesting that the antigenicity of the F glycoprotein on HVPs was well preserved. These results also suggested that HVPs cannot enter host cells using the IAV-HA protein and likely enter via the RSV-F protein instead. To test this hypothesis, the authors treated cells with neuraminidase, which binds sialic acid and sequesters it, thus leaving no receptor for IAV to bind to and enter the cell. Viruses isolated from singly or co-infected cells were then used to infect these neuraminidase-treated cells, and the cells were stained with IAV and RSV nucleoprotein-specific fluorescent antibodies and visualized by fluorescence microscopy to determine whether IAV, RSV, or HVPs had infected them. As expected, neuraminidase-treated cells infected with viruses isolated from RSV only or IAV only infected cells contained RSV nucleoprotein but not IAV nucleoprotein, suggesting that IAV was unable to infect these cells because there was no sialic acid to bind to, while RSV infected these cells normally because RSV entry is not dependent on sialic acid. Interestingly, neuraminidase-treated cells infected with viruses isolated from co-infected cells contained an abundance of IAV nucleoproteins, further implying that HVPs containing IAV genomes entered these cells using the RSV F protein. To confirm that the RSV F protein mediated HVP entry into cells, viruses isolated from co-infected cells were treated with a monoclonal antibody against RSV F protein before they were used to infect neuraminidase-treated cells. The monoclonal antibody would presumably sequester any viruses having the F protein and prevent any F protein-mediated entry into cells. This treatment led to significantly reduced entry of HVPs into cells, confirming that the RSV F protein mediates entry of hybrid particles into cells. Studies of virus-host interactions are extremely common, and scientists have made a lot of progress in understanding the mechanisms that drive these interactions. In stark contrast, we know very little about how viruses interact with each other. Some work has shown that not all co-infections are successful and often result in competitive exclusion, with one virus displacing the other, thereby preventing it from completing a replication cycle or establishing an infection in the first place. To my knowledge, this is the first study showing that two completely different viruses can coordinate their replication cycles to develop some kind of symbiosis in a clear display of coevolution. And although this phenomenon may seem extraordinary, it is probably more common than we think. That's it for today's episode. Thanks again for listening, and don't forget to subscribe. My name is Trudy Ray, and I'll see you next time on Catch This.